Sorry. So last day we talked a little bit about how food structure can actually change the dentation of our teeth. So the size of our teeth changes in response to cooking of food. So the first couple days, what I want to talk about is a little bit of an introduction into food science for the first 5 or 10 minutes, but then I really want to focus on what happens in the human body when you are exposed to food. So what happens when you see food, what happens when food enters your mouth, your stomach, and I want to look at it both from a nutritional aspect, so how does a low glycemic versus a high glycemic food respond, but also how does processing your food change the glycemic index. So if you refine a grain or if you gelatinize a starch, how does that change the bioaccessibility of the macronutrients? So the first few lectures are going to give us the background we need to understand how the body responds to food. So for the people in here who are food scientists, and even nutrition and anyone who deals with food, it is an extremely challenging feat. And the reason for that is we're really a combination of 4 disciplines. You could be a chemist, a microbiologist, an engineer, and a nutritionist. And one of the challenges we have in academia is we treat those disciplines as individual silos. So as a food chemist, I sit in my office and I work on trying to create new and novel foods. And I often don't do that in collaboration with a microbiologist who's going to try to understand the safety of the microbial hazards of those changed foods. Then there's not a processor looking at them. So does that food or does that chemistry change how we have to engineer that food? And then, on top of that, how does all those changes influence the healthfulness of that food? So when we talk about chemistry of foods, we're talking about things like adulteration. So are you buying what you think you're buying? So right now, there's a huge problem with 2 different things. The first is fish. When you consume fish, there's a high incidence right now of people selling low quality cuts of fish and trying to pawn them off as more expensive cuts. So once you've removed the head, removed the skin, it's extremely difficult to identify what species of fish that was, especially for an average consumer. So are you going to the grocery store and are you buying, let's say, a tilapia, or are you buying a less expensive pollock fish? So chemistry allows us to understand the molecules that are in that food and that we can use as a fingerprint to identify if that food is indeed what it says it is or not. Another example is Kopi Luwak coffee. Has anyone ever heard of Kopi Luwak coffee? A few people? So Kopi Luwak coffee is a coffee that's been eaten by a civet cat. So it's a small vermin-like animal, and then the feces of that animal is collected because it gorges itself on coffee beans, and then we sell that coffee for extremely high prices. So because the digestive enzymes of that civet cat have actually caused that coffee to undergo proteolysis. So it's breaking down that protein and it's making a much, much more subtle flavor. But what will happen is people will put digestive enzymes on coffee and then sell it as Kopi Luwak coffee. So they take coffee, which is a couple cents a glass to manufacture, and are now selling it for $10 a cup. So this is some aspects of food chemistry that we really need to understand what is in our foods so we can ensure that we're actually selling the appropriate thing. Microbiology is making sure our foods are safe. And this is extremely challenging. So when we talk about microbiology of foods, there's never a sterile food product. So we often talk about the process of sterilization in foods, but sterilization involves killing all bacteria that's present on that surface. So when we talk about things like medical instruments, or if you get a tattoo, sterilization is done on the needles, because we can heat that metal to extraordinarily high temperatures for very long times, and we're not going to do anything to the metal needle or the syringe that we're going to inject you with. But in foods, if we undergo sterilization and kill every bacteria that's present in foods, that food will very quickly become inedible. So we never try to get foods sterile. We try to kill pathogens so you don't get sick, but more importantly, we try to limit the growth of pathogens by creating barriers to growth. So if E. coli is in your lettuce or is in your hamburger meat, we can then process it, to get as much of that E. coli out so you don't get sick, but there still may be spoilage microorganisms present. So in pasteurization of milk, that milk is safe to drink. There's no pathogens present. But if you leave that milk on your counter for a few days, it's going to spoil, right? So there's still bacteria present in that milk, just not pathogenic microorganisms. When we talk about food engineering, so how we process foods, how are we going to make sure that raw milk, which is going to contain things like salmonella, E. coli, the virus or the bacteria that causes tuberculosis, it's all going to be present in that milk. So how do we put it under a unit operation to actually minimize or reduce the spoilage microorganisms and eliminate the pathogens? So we have to be very, very familiar with what inhibits microbial growth. Things like water activity, pH, salt concentration. So by modifying those parameters, we can then inhibit the growth of microorganisms in food. But we have to be able to look at the chemistry, so the sugars, the salts we're adding. We have to understand how that microorganism grows. We have to understand how to process that food to ensure that there's no pathogenic microorganisms there. So are we going to pasteurize it? Are we going to can it? Are we going to use a high pressure processing or something that's novel that doesn't undergo changes in the organoleptic properties? So if you talk about pasteurized milk versus raw milk, if there's anyone in here that grew up on a farm, they're always going to, or on a dairy farm more specifically, you'll often hear adages that raw milk tastes better. Well, raw milk hasn't undergone the Maillard reaction. So when you pasteurize milk, it undergoes the Maillard reaction. So if you compare raw milk with pasteurized milk, you can actually see the pasteurized milk is a little bit brown because of the Maillard reaction. This changes the flavor profile, and people can actually detect those changes in the sensory properties of milk. So we have to figure out the best way to process a food to ensure that the microorganisms are unable to grow in that environment, but are still stable, they're still desirable to consume, and they're still nutritious. So when we talk about the engineering, the microbiology, the chemistry, we then have to apply all of that to understand how to formulate new foods. Because there's all kinds of amazing things I can do with foods. I can create neat gels and neat protein matrices that mimic meat. But if they're not shelf stable, if they're not nutritious, and if they don't have a specific shelf life, what good are they? So we have to make sure that these technologies are integrated in all four aspects. So one of the challenges when we talk about a food science major or someone that's working in the food industry is you're not getting a single degree. You should actually be getting a degree in microbiology, a degree in chemistry, a degree in engineering, a degree in nutrition. So it's a very, very complex field that requires a lot of integrations of different disciplines. So nutrition has changed a lot in the last 200 years. So when we talk about classical nutrition, this is understanding that certain foods prevent specific diseases. So 200 years ago, we knew that lemon juice and orange juice prevented scurvy, right? We didn't know why it did that, but we knew if you drank orange juice or lemon juice, you would be fine and you wouldn't develop scurvy. So on long voyages, which we'll talk about later on in the course, we'll talk about the Franklin expedition. So the Franklin expedition is when we tried to sail the Northwest Passage. They put lemon juice on the ship, barrels and barrels and barrels of lemon juice to ensure that the sailors didn't get scurvy. But that was our understanding of nutrition at that point. So then we started to develop this idea of optimal nutrition. So optimal nutrition is where we get the right nutritional compounds to prevent or to control disease. So this is things like understanding certain fatty acids have a beneficial role in cardiovascular health. So your omega-3s, your omega-6s, your omega-9s. While other fatty acids, trans, let's say, or really debatable, but let's say some saturated fats, are not cardioprotective. They actually have a detrimental effect on cardiovascular health. Now we've moved into this era of molecular nutrition. And molecular nutrition looks at specific molecules and how they interact with specific metabolic processes. So for example, if you have compound A and it's going to be an antioxidant, they will show it interacting with a pathway in the human body that provides evidence that it actually behaves as an antioxidant. And this is where nutrition is going. And what's really exciting about this area is every single one of us have a different biological makeup. That means that every one of us is going to interact differently with our food than the next person. So person 1 can eat a cracker, and person 2 can eat that same cracker. Now, depending on where you are in a phylogenetic tree, or what your evolutionary path was, you will produce different amounts of amylase. So we have replication of amylase gene numbers. So some of you are super producers of amylase and will digest carbohydrates faster than other people. Does that make sense? So everybody's biology is made up a little bit differently so that our response to food is unique. So this introduces a concept of personalized nutrition. So if I'm an academic, and obviously I'm somewhat sedentary, so I don't move a lot, I'm fat. So because of that, my nutritional requirements are very different than a marathon runner, or a child, or a woman, right? So we all talk about folic acid fortification. Folic acid fortification, does anyone know why we did folic acid fortification? Where do we fortify folic acid in Canada? Breakfast cereals. So all breakfast cereals in Canada are fortified with folic acid. Does anyone know why we fortify cereals with folic acid? Yes? Right, so to prevent neural tube defects when we're pregnant. Do I need folic acid? The same as a pregnant female would need folic acid. No. The other thing too with folic acid is all early chemotherapeutics worked on folic acid metabolism. So if you have, let's say, an early prostate cancer, so a 50-year-old male, early prostate cancer, folic acid is not good for them. Folic acid increases cell division, and if you have cancer, that is not a good thing. So again, it's introducing the idea that everyone's nutritional requirements are a little bit different, depending on what their performance is tailored to. So now we're into this whole era of personalized nutrition. This really is really important and really prominent in athletics. So if you're a marathon runner or a football player or a soccer player, this concept of personalized nutrition is extremely, extremely important. So a lot of this course is going to deal with the first 3 macronutrients: carbohydrates, lipids, and proteins. How do they break down in the human body? But also, how can we develop structures of those products? So for lipids, let's say, when you eat a good chocolate bar, so a really high quality dark chocolate, you put it in your mouth, you leave it there for a second, and it begins to melt. That fat coats your tongue, you get a nice sensation of flavor in your mouth, and it melts down, and it's delicious. Now, if any of you grew up with parents who didn't like buying, let's say, Easter presents and bought you that really cheap chocolate, so that really cheap hollow chocolate, you put it in your mouth and it doesn't melt. The chocolate's the same, but why doesn't it melt? Because of the polymorphism, or how those crystal structures actually arrange in 3-dimensional space, change how that fat melts and behaves in your mouth. So we'll talk about the chemistry, the design, the engineering of these different macronutrients. How can we create novel food structures? How can we create healthy food structures? How can we create food structures that induce satiety, so you feel full longer? By understanding the chemistry and the food science of that, we can actually begin to tailor foods so they break down in specific ways, so you feel full longer, so you eat less food, to try to combat the whole problem of obesity in Western society. So when we talk about foods, our interactions with food is extremely complicated. It's a social aspect, right? We desire foods. We build family events around food. Thanksgiving, we all get together and celebrate around food. But then how does it behave in the body? We make our very first influence, our very first opinion of what we're going to eat by visually looking at it. Is it bruised? Is it overly ripe? Is it going to be safe to eat? Those are all visual cues. We smell the food. We understand, even before we touch it, whether or not that food is going to be somewhat safe to consume. We then put it in our mouth. This is where we make our next set of sensory observations about that food. How does that food break in your mouth? Is it chewy? Is it sticky? Does it shatter when you break it? All of these things refer to the ingestion. So how do you form a bolus? How does that food break down in the oral cavity? Digestion happens through a couple points in the digestive tract. So it happens in the oral cavity with the release of enzymes. So specifically we have salivary amylase, so we can break down carbohydrates. Lingual lipase, so we secrete enzymes that can break down fats in our mouth. They then go into our stomach, and again, acid is released, pepsinogen is released. So pepsinogen is a proteolytic zymogen, so it's going to break down proteins. And then it goes into the small intestine, and it's further broken down by pancreatic enzymes. These nutrients that are broken down, so glucose, fructose, I'm missing one, glucose, fructose, and what's the other monosaccharide? Sorry? Galactose, that's it, thank you. So those three sugars can be absorbed by the body. We can absorb fatty acids. We can absorb 2 monoglycerides, so a glycerol with a single fatty acid backbone. We'll talk about these in a minute. We can absorb amino acids. We can absorb very, very small peptides up to a maximum of about 3 amino acids. And then our body reassimilates them. So they take those amino acids, they take those sugars, and they use them as building blocks to build back the aggregates, the lipids, the proteins, the carbohydrates. And then we expel that food. So we poop it out. And we do this through a complex series of organs. So we have our primary organs, our mouth, our stomach, our small intestine, and our large intestine. Then we have the oscillatory organs, which play a role in digestion, but the food never comes into contact with it. So the pancreas releases enzyme. The liver produces bile. We have the gallbladder, which stores bile. And we have the kidney that filter out our waste products. And all of this is regulated within very, very fine limits. And this is all done through homeostasis. So homeostasis, there's all kinds of different homeostatic pathways we have in our body. We have our cardiac rhythm. So while we're tired at night, certain hormones are released in the evening, others are released during the day. We have mechanisms that control satiety and obesity. And we're going to talk about this in a lot of detail over the next 3 days. So we'll talk about enzymes like ghrelin, the hunger hormone, or leptin. So if you get obese and you produce a lot of fat, you produce a lot of leptin. So what drives us to eat? There's short and long-term effects when it comes to satiety, both feeling of fullness and feeling of hunger. So the short term, we get hungry. It's our appetite. This is our psychological drive to eat. So if you're out with friends and all your friends are eating, you may have a higher higher likelihood of consuming food because everyone around you is eating. Or if you're at a Thanksgiving dinner, because there's a whole bunch of choices, you may fill your plate up with more food. Plate size, the size of a bowl, the color of a bowl, the color of the plate, the number of people you eat with when you're eating, all affect how much food you actually consume in a single sitting. So it's not just the hormones and the neural transmissions that drive you to eat. There's a psychology that drives you to eat as well. So this is our appetite. Again, this is influenced by environmental factors. There's cues that we don't even understand that will trigger someone to eat. So if you see something or smell something you like, it all of a sudden makes you more and more hungry. We have hunger, which is our biological drive to eat. This is transmitted through hormones, neural transmissions, and cytokinins. So this is when your blood glucose drops, a whole bunch of hormones and enzymes are released by all along your gut-brain axis. And what that means is from your brain to your stomach, it's connected with a series of nerves and organs. All of those will begin to produce different hormones, which will trigger you to be hungry. It'll start your stomach growling. You'll start to get moody or hangry, right? All of these things are actually hormones triggering your body to become more hungry. Now, what drives us to stop eating? So we've sat down, we've had our hunger triggered, you're eating your meal. Why do you stop? You get the perception of fullness. So the distension of your stomach gets full. You begin to release hormones. We're going to talk about CCK and PPY in a couple minutes. So your stomach fills up, you get distension, and then you get a cascade of hormones. And these hormones interact all over your body doing different things. They're telling your body, we've just eaten, let's start to secrete enzymes. Or let's slow down that food moving through our gastric tract. So our stomach begins to contract more slowly. Our intestines begin to push that food more slowly, so we have more time to digest it. And then we have this important concept called the ileal brake. So when food starts to leave your stomach, enters your duodenum, moves into your ileum, more hormones are triggered. These hormones then have feedback mechanisms to say, you're full, you're done eating now, let's start to produce enzymes, let's start to digest. So all of this behavior is influenced also by the hormones, the neurotransmissions, and the cytokinins that your body releases in response to food. And in response to where that food is specifically located in your body. And satiety is your perception of fullness after a meal. So if you go and you have a liquid smoothie with 1,000 calories, or you sit down and you eat that same 1,000 calories, depending if you drink it or if you eat it, you're going to feel full for different lengths of time. If you have a big salad versus a bowl of soup, that length of satiety is going to differ. So we talk now a lot about dietary fibers are great at inducing satiety. You feel fuller longer. So you either have a delay in consuming your next meal, or when your next meal comes, you actually consume less food. Satiety based on certain parameters of that food. So what is our biological drive to eat? Our biological drive to eat is driven by a series of nervous impulses, cytokinins, so immune system, and our endocrine system, so a series of hormones circulating. And we'll talk about these at different points over the next 3 or 4 days, because there's a whole bunch of them. So the first ones are biological drive to eat. So these are our neuropeptide Y family. The neuropeptides, they're triggered when your blood glucose drops. So when you have low blood sugar, your brain all of a sudden says, oh no, the only energy source I can utilize is glucose. So it doesn't matter how much fat you're consuming, doesn't matter how much protein you have in your blood. If you don't have glucose in your blood, your brain cannot function. So we have to release a set of impulses. So neuropeptide Y family is this first initial cascade. It's the first hormones that are released when your blood sugar drops low. So what does it do? Well, neuropeptide Y is released all along the gut-brain axis. So it's released in the brain, it's released in the gastric compartment, it's released in the intestines. It regulates energy homeostasis, so how we're actually metabolizing that energy that's present. It increases gastric motility, so your stomach starts to gargle. You start to feel hungry, your stomach starts to churn. It also has a role in inflammation. So the next set of hormones are your PYY. This is released postprandially. So after you've consumed a meal, PYY is released from the intestinal tract. It tells your GI to slow down, to let that food have a high residence time. It needs a high residence time so those enzymes can break down those macromolecules into monosaccharides, into amino acids, into free fatty acids, and into monoglycerides. This allows then more absorption of those nutrients. It also triggers satiety, so it triggers you to stop eating. And there's these papers that I put on, they're really cool, and they show where or how they know that these hormones actually affect satiety. So in this study here, they were able to take PYY and put it in peripherally, so inject it into the person, and then they compared how much food two different people ate if they were administered PYY and if they weren't. And the other really interesting aspect of this is PYY is actually produced by your gut microflora, by specific bacteria. So imagine, everybody in this room has a very, very, very different gut microflora. Everyone has about 400 to 500 different bacteria, species of bacteria, and the populations of those different bacteria influence our satiety. So they can actually interact with our neural transmissions, with our hormone systems. It's remarkable, the host microbiome interaction that exists in our body. And we'll talk a lot about that in the coming days. PP, which is pancreatic polypeptide, is primarily secreted in the pancreas, a little bit also in the intestinal tract, stimulates the vagus nerve, and again, introduces or induces satiety. And how do they know this? Children with a genetic defect with Prader-Willi syndrome actually don't produce PP, and they are clinically typically obese. So they can make these observations. So depending on the macronutrient that's present in that food, if it's fiber, if it's fat, if it's carbohydrates, this changes our long-term satiation, our short-term satiation versus our long-term satiety. So if you eat a meal high in fiber, a salad, you can't eat a lot of it, you feel full very quickly. But very quickly after the meal, you're hungry again. So again, satiation is the short-term influence. Satiety is the long-term influence. And a great, interesting read if you're interested. But it's not that simple. Noaz, can you give me a hand? Can you just unplug that for a sec? Yeah, grab a thing of bread. Here, grab a thing of bread and come with me. No, the whole box, right here. No, carry it up with me. Take off the top layer, the top thing. All right, let's go. So we'll take a nice time and we'll see what begins to happen. So fresh baked bread just came from with the grain from downtown Guelph. Keep moving up. What's happening? Keep going. So we're actually triggering right now a cephalic response. Keep moving. Oh, how are we doing with the power cord? I'm out at full extension here. So we'll just walk it around. So just being exposed to the idea of food, the smell of food, the sight of food is enough to trigger a cephalic response. So what is this cephalic response? So the idea of food, you think about it, you see it, you smell it, you touch it, it doesn't enter your mouth, and already your digestive tract kicks into high gear. I am so hungry right now. So it stimulates your cerebral cortex. Your brain starts to think about food. It triggers your hypothalamus, your medulla oblongata. All of a sudden it sends a neural impulse down your vagus nerve, and your stomach begins to do stuff. So right now, at the smell of that bread, all of you are beginning to produce gastric acid. Why? Because you're preparing to consume food. Why are you producing gastric acid? Well, gastric acid, so gastrin stimulates the perineal cells. These begin to produce hydrochloric acid. The pH of your stomach begins to drop. It also stimulates your chief cells to release pepsinogen, which is an inactive precursor to pepsin. So pepsinogen, if you think about it, will degrade proteins. What is your biological material made of? Your cells, your muscle. It's all protein. So if your chief cells begin to produce pepsin, it's going to start to metabolize itself. It's going to start to cause cellular damage. So we create these inactive enzymes called zymogens, and these zymogens can be released safely by the chief cells, move into the gastric phase, interact with hydrochloric acid, and all of a sudden, pepsinogen becomes pepsin. Why am I, sorry. No, Go back. So when you see foods, again, the idea of this cephalic response is your body begins to produce enzymes and hormones that begin to interact or release at the sight, the thought of food. So just talking about food, you can actually measure changes in the gastric secretions. So you can put a probe into someone's stomach, sit down beside them, and talk about food, talk about the cultural aspects of food, how good something is, and all of a sudden their stomach begins to produce acid. This happens at the sight of smell. Sorry, at the sight of food. The smell of food and the sight and smell of food, it begins to release acids and hormones that are there ready to digest that food when it comes along. So the first question, did the idea or the smell of bread trigger anyone's cephalic response? Does anyone feel hungry now after being exposed to that bread? Don't start eating it right away. So the first organ that interacts with food is the mouth. So the first thing I want to talk about is the idea of oral food processing. Do you want to pass these around and take a crack or two? I'll start these ones at the back. You want to just take a cracker and pass them around too? Thanks. Yeah, you can just pass the box. Okay. So, you can start eating. I want you to be conscious though of when you bite into it. How much force does it take to rip that bread? When you chew it, how much force does it take to actually compress that bread with your molars? And then when you release it, the springiness of that bread. All of those things are sensory cues to quality. You'll say a bread is high quality if it's soft, if it's got a good recoil after you bite into it. Now, the other thing you do in your oral cavity is you release a series of enzymes. So if you want, take a bite of the bread and leave it in your mouth for a minute. You'll notice the sweetness actually increases. Why is the sweetness increasing? Because salivary amylase is beginning to break down those carbohydrates into maltose. Maltose has a very mild sweet sensation. So if you leave something high in carbohydrates in your mouth for a long time, you'll actually begin to detect the sweetness. Now, what we're watching here is the formation of a healthy bolus. So I'm going to rewind it. Okay, so this is what you call a barium swallow. So a barium swallow is a contrasting agent. So if you look at the mouth, my pointer's not working, but you see that really dark thing go down the throat? So this is what they call a barium test. So barium is what they call a contrasting agent and picks up really easily on an X-ray. So if you watch, so here's a barium-enriched liquid. You notice that really smooth transition down the throat. So I wish my pointer worked. Actually, I can use my mouse on my thing. So I want you to watch right in here. So this is a really, really important phenomenon. So your trachea and your esophagus are connected. So when you swallow, your epiglottis has to close your trachea. If it doesn't close your trachea, the food will enter your airway. And we'll talk about why this becomes very problematic. So what you're watching right now is a healthy individual consume what they call a barium-enriched food, so a contrasting agent. We're going to talk about taste, but we're going to talk about it in a little while. So we're going to jump the whole idea of taste. Just put a little advertisement for the little bakery downtown that supplied us with the bread. So, oral food processing. This is how you perceive a lot of food. So when that food is in your mouth, you are making decisions about whether or not it's quality or it's not quality. So when you bite that piece of bread, you'll notice it fractures into two pieces, right? You bite it, one piece turns into two. Not an overly exciting break profile. Take your cracker now and bite that cracker. When that cracker breaks, all of a sudden it shatters. So the oral processing and the sensory cues that you're using to decide whether or not something is good quality is different for a cracker versus a bread. So when you bite that cracker, it shatters into a lot of pieces. It then takes different amounts of energy every time you bite that cracker, because all the mucus in your mouth is interacting, is absorbing into that food, it's softening that food. So there's two major things that are happening right now while you're eating. The first one is you're reducing the size of the particles, right? So obviously you're limited by the diameter of your esophagus. Later on in your stomach, you're limited by the size of your pylori sphincter, which we'll talk about. So foods have to be a certain size to get through your digestive tract. So you're decreasing the particle size of that food. You're chewing, you're breaking it up. The other thing you're doing is you're lubricating it. You're adding salivary liquids that are incorporating into that food product, that are moistening it. So there's two different platforms or two different requirements for oral processing. The first is reducing the particle size. The second is lubricating it. You can't swallow if either of those aren't in place. So if you ever take, Fun Dips, it's like that sugary stuff. If you're ever bored, buy one, pour it all into your mouth, and then try to swallow it. You have a fine particle, right? It's small, it's granular, it should just go right down. But because it's not lubricated sufficiently, you can't swallow. So swallowing and that formation of that bolus is dependent on the size of those particles, but also on how lubricated that material is. And that whole area of study is called oral food processing. So it starts from your first bite fracture. So what happens to that food? Does it break into 2 pieces? Does it shatter into 1,000 pieces? Then you have to chew it. You have to masticate it. You have to break down the particle size. All the while, you're secreting enzymes, you're secreting saliva, and you're lubricating that food. You're forming a consistent bolus, which we saw a second ago, can then pass past your epiglottis, move into your esophagus, and reach your stomach. We take a lot of cues based on that oral processing. So imagine taking a Ritz cracker or a Premium Plus cracker and biting into it and it being soggy, right? We very quickly realize that that's not desirable. So we make our first sensory cues with how our teeth interact with that food. But while we're masticating and while we're lubricating it, we're releasing 2 enzymes as well. The first enzyme is salivary amylase. Salivary amylase can break the bonds, and we'll talk about the types of bonds later, can break the bonds between alpha-1,4 glycosidic bonds. What are in linear, glycemic just means digestible, carbohydrates. So when salivary amylase is released, again in the oral cavity, it is able to cleave those long chains into dextrin, so short chains of sugars. It can produce maltodextrose, and disaccharides. It cannot be reduced to the monosaccharide in the oral cavity. We only produce amylase, which can only break down amylose, amylopectin, to the smallest subunit, which is maltose. So this is why if you leave a carbohydrate in your mouth for a long time, you get a little bit of sweetness. So if you leave a piece of bread in there for 5 minutes, I don't know if it's 5 minutes, a couple minutes, you'll notice the sweetness of that bread increases. It's because you're actually monitoring the amount of salivary amylase, and that's cleaving off maltose. The amount of maltose is able to trigger a sweetness perception. Now we also produce lingual lipase. And I'll be honest, my knowledge of enzymology is somewhat low, but lingual lipase and the lipase family are fascinating. Why do I say that? Well, first, lingual lipase remains active in the stomach. Now, why is that interesting? Most proteins denature in acidic environments, right? So if a protein or an enzyme, same thing, if an enzyme unfolds it loses its catalytic activity. So when you denature a protein, it no longer is biologically active. So why do we thermally process tomatoes? To reduce enzymes that break down pectin, so it stays nice and thick in your seltzer and your tomato juice. So lingual lipase is made up of 3 amino acids: aspartic acid, histidine, and serine. And this is what we call a catalytic triad. Now, it's fascinating. This catalytic triad undergoes a series of deprotonations, and what it does is it creates, the aspartic acid loses its hydrogen. This causes a set of electron shifts and makes that oxygen more electronegative. Why is that important? For anyone who's taken first-year chemistry, a good nucleophile is something that's able to donate electrons to another molecule, right? You talked about this in first-year chemistry. If you're taking second-year orgo, you'll talk about it again. But these 3 amino acids work in a cascade to create this triad that allows that hydroxyl group to attack a carbonyl in a lipid. So what happens then is that oxygen undergoes nucleophilic addition and cleaves off a fatty acid. Now, why is this important? Well, you can imagine an enzyme is this globular protein, right? And this globular protein contains this catalytic triad somewhere within that enzyme. That's called the active site of the enzyme, where that catalytic triad is. So now we have a triglyceride. That triglyceride has to arrange itself into that catalytic site. It has to sit in that catalytic site, and then that hydroxyl group has to position itself to a carbonyl to break that carbonyl bond in the lipid. Now, when we think about that, when we think about the catalytic site is going to have certain geometric constrictions. So if it's a sphere, it's going to be hard to fit a square into it. So the catalytic site is going to determine if a long-chain fatty acid can reside in it or not. The other thing that's really important is this catalytic triad can only interact at the ends of the molecule of lipids. So it can only cleave off the fatty acids at position 1 and position 3. Position 2 is very, very rarely digested by lipolytic enzymes. There's only one example that I know of where you can actually cleave the position 2. And it's in maternal lipase, which is found in breast milk, which is fascinating in itself because breast milk will actually convert a triglyceride into 3 fatty acids and a glycerol. All other human lipases convert it to 2 fatty acids and a monoglyceride. So why would maternal lipase, or lipase found in breast milk, have a different activity? Well, because at an infant, your stomach pH doesn't drop to 1.5, it stays at about 3.5. Plus, your pancreas isn't producing all the enzymes. So your body, as a newborn, as an infant, really relies on its mother's enzymes to actually begin to break down the nutrients. And this is extremely important in infant nutrition because when that baby is born, it goes from relying 100% of its energy from glucose to the vast majority of its energy coming from lipid sources. So the ability of that fat to be digested is extremely important. So maternal lipase, if you're talking about a breastfed versus a bottle-fed or a infant formula-fed baby, there's differences in how that baby or how that infant interacts with the lipids. So how that energy is metabolized and used changes depending on if you're formula-fed or if you're breastfed. And now they're talking about, are there links to obesity? There's definitely changes with your gut microflora, depending on if you were bottle-fed or if you were fed from the breast. So these all have extremely important consequences for a lot of other things. Now, what's fascinating, again, is these are cleaved to produce, salivary amylase cleaves the fats to produce 2 fatty acids and a 2 monoglyceride. Okay? So we are able to absorb those. Now, when we talk about this catalytic triad, what's absolutely remarkable about it is once you cut off that fatty acid, so once I've removed that bond between the carbon and the oxygen, that oxygen needs another hydrogen. That catalytic triad can actually donate its hydrogen to the glycerol, or to the monoglyceride, to complete the reaction. So we're not left with this radical that exists. So we have the nucleophilic addition onto the carbonyl. That cleaves the double bond. The histidine then donates that hydrogen to the position 1 or 3 position, wherever that fatty acid's been cleaved, making it a complete monoglyceride. Now, I want you to think about this for a second. A typical enzyme is 300 to 500 amino acids. If we randomly were to put those amino acids together, just think about the number of combinations, the permeations and combinations of that. 300 to the n prime, right? Now, imagine the number of different orders we can put them in, but now imagine every time we create a position. So amino acid 1 in position, in relative to position to amino acid 25. This is how that protein folds. This is the secondary structure. We talk about alpha helices, beta sheets in first year biology, hopefully. How those amino acids arrange in three-dimensional space is extremely important. This catalytic triad has an amino acid at position 203, another amino acid 54 away, and another one that's at 144. So you have these 3 amino acids that have to be together to create this catalytic triad that's going to create that nucleophilic serine, that hydroxyl group that really wants to donate electrons. You've got this histidine that's holding a hydrogen for the reaction, saying, once you're done, I'll give this up. And they're not even beside each other on the amino acid backbone. So think about the complexity. You have this protein that's folded into secondary structures, into ternary structures, into quaternary structures, which we're going to talk about in the protein section. And the way they fold, those 3 amino acids end up in close proximity and are able to catalyze reactions. The biology of it, the chemistry of it is truly remarkable. If you were to just let this happen and take random selection and hope your enzymes work, it would be like 1 in a trillion enzymes would have activity. So why does our body produce enzymes with such specific configurations? How does it know to pack into an alpha helix? And how does it know to fold 2 alpha helices together to put those amino acids into close proximity? Again, it's remarkable that this is able to happen. And this catalytic triad exists in all different enzymes. It's in proteases, it's in gastric lipase, and we'll talk about them again and again. So lingual lipase is secreted in the oral cavity, doesn't have a lot of activity. It really needs to be in a more ideal pH, so it's not going to create a lot of lipolysis in your mouth. So it doesn't require bile salts or colipase, which we're going to talk about in a little bit. It hydrolyzes medium chain triglycerides. So again, that catalytic site is restrictive. So long chain fatty acids have a harder time arranging themselves into that catalytic site. So then that catalytic triad of aspartic acid, histidine, and serine can't get at that carbonyl group. Now, what's really interesting is that gastric lipase and salivary amylases play a very small role in digestion. They're important. They're extremely important when we talk about infant nutrition. So if we don't talk about, for most of us, lingual lipase doesn't play a significant role in digestion. But if we talk about an infant, or especially a preterm infant, lingual lipase and maternal lipase are the 2 most important. So when you're designing an infant formula, how do you design an infant formula that's able to undergo very fast lipolysis? So if you have anyone that's recently had a child, some of the challenges with breastfeeding is a lot of times the baby won't, what's the word I'm looking for? Not latch, it'll reject the breast milk. This is with mothers that overproduce amylase. So if you actually overproduce amylase and lipase, and break down that lipid, it gives you a very soapy sensation. Fatty acids, they're not pleasant tasting. So mothers that overproduce maternal lipase will actually often have a harder time breastfeeding because the baby rejects it. It doesn't like that soapy fatty acid taste. But again, when we talk about infant nutrition, we really have to be conscious of lingual lipase, maternal lipase, gastric lipase. For adults, lingual lipase doesn't play a major role in digestion. What's interesting again about maternal lipase, it is able to cleave all 3 fatty acids off the glycerol backbone. In adults, no enzyme is able to remove the fatty acid from the SN2 position. So that middle fatty acid is not cleavable. We absorb it as a 2 monoglyceride. And what's interesting is there's enough maternal lipase in breast milk to fully hydrolyze that meal in about 30 minutes. So the baby doesn't even need to produce its own enzymes. The mother is actually supplying it through the breast milk herself. Now, so we've talked about the size of the particle reduction of foods. We've talked about oral lubrication. We've talked about the 2 digestive enzymes. Now we have to talk about swallowing and the formation of that bolus. So we've talked about bolus formation, and for probably everyone in this room, the ability to form a bolus is nothing we even think about. In senior citizens, especially senior citizens that have had a stroke, there's a condition called dysphagia. And dysphagia is a difficulty swallowing thin liquids, a glass of water. So that's just a Twitter poll. Has anyone ever witnessed a grandparent who has a glass of water and coughs continually after they consume water? Yes, no, I don't know. So again, when your grandparent consumes a glass of water, does that water cause them to cough? So, about 1/3. And what's really cool about that, that's about the percentage of the population in senior citizens that are dysphasic. So what is dysphagia? So the first minute of this are healthy swallows. So again, this is a thin liquid, something like water, milk, barium, which is the contrasting agent, and we're watching it go down. So this video is a little bit overexposed, but if we watch, I want you to focus right where the cursor is, right in this area. So in dysphagia, when you swallow, your epiglottis has to close. This then creates a continuous tract down the esophagus, and the stomach then enters goes down the esophagus and enters the stomach. With dysphasic people, or people with dysphagia, that epiglottis doesn't close properly. And we'll see it in a second what begins to happen. Just wait for it. Wait for it. Still waiting for it. So again, the darkness of that fluid is associated with this large molecule called barium. Barium is a contrasting agent. It's radioactive, so you don't want to do barium swallows often. We do barium swallows at the University of Guelph often to understand how people swallow and interact with food. What's really interesting about dysphagia is it's actually typically diagnosed by speech pathologists, which is mind-boggling to me. It just seems like a very strange, adult abnormal swallow. So here we go. I want you to focus now at the back of the throat where the epiglottis and esophagus, where the airway, right in this area here. So watch right here where the mouse is. You're going to see a small amount of fluid move forward into the airway as opposed to going down the esophagus. So you can see the fluid is actually kind of built up and stuck in the back of their throat. And then when it opens, there we go, you can see it going down the esophagus, the majority, and that small amount going into the lungs. This causes aspiration pneumonia. So when you watch your grandparent taking a glass of water and coughing, it's because some of that water has actually went into their airway, and it's one of the leading causes of pneumonia in our senior citizen population. So it's extremely, extremely important. In most severe cases, it causes choking, but typically it results in the development of pneumonia. At the University of Guelph, we have one of the world's leading experts in designing foods to prevent dysphagia. So if you're dysphasic, you have a hard time swallowing thin liquids, water, things that are not viscous. So she studies how adding specific hydrocolloids changes the thickness, but also changes that response. So can we mitigate dysphagia by changing the chemistry or the material properties of that food? The answer is we can. It's very difficult to do, but you can actually read about it with modified texture foods. Now, so we've talked about the oral cavity. So enzymes released, the two primary enzymes are amylase and lipase. We have lubrication. Not only do we have lubrication in the formation of the bolus, we also have the particle size reduction. So the particle size reduction and the lubrication allows us to form that bolus, allows us to swallow that food. That food then reaches the stomach. Now, the stomach is broken into four main categories. The cardia, the fundus, and the atrium are all secreting mucin. Now, mucin is extremely important because it has to line the stomach. Why does it have to line the stomach? Because again, proteases are active in the stomach. So if that protease and that cell or the stomach come into contact, it's going to begin to hydrolyze itself. It's going to cause ulcers. So this mucus protects the lining of your stomach from the digestive enzymes and that acidic pH that's naturally present in the stomach. We've got these thick folds, and these folds are secreting acids and pepsinogen. Again, pepsinogen is the zymogen. A zymogen is an inactive precursor to an enzyme. So a zymogen needs to undergo some biochemical change to become active. In the case of pepsinogen, it's just in an acidic environment, it removes a portion of the molecule. It then goes from the zymogen pepsinogen to the active enzyme pepsin and can start breaking down amino acids. We also have the pleura sphincter, which we'll see in a minute, which is a very small opening at the end of your stomach, which leads to your intestines. This regulates the size of the particles that are able to go from the stomach to the duodenum. Now, we typically subdivide the stomach into two primary areas. The fundus is the storage compartment. So that's, once you've eaten your bread, that's where your food sits until it can move to the distal stomach, where it actually is interacting with the gastric acids, but also those antral contraction waves, which we'll see in a second. So your food enters the stomach. Now, your stomach is very small, a few centimeters when it's not full. So you consume food, and if you were to consume food with no neural impulses, every one of you would be regurgitating now, because your stomach has to expand. So when the food enters your stomach becomes distended, and you get this vagal, I can't say it, I'm not going to try, causes distension of the stomach. That causes the vasovagal reflex. This goes up the vagus nerve, tells you I'm full. It also releases acetylcholine, which causes your stomach to relax. So you're not going to sit there and your stomach's not going to recontract to its normal size and expel the food that you've just eaten. What else does it do? It triggers the perineal cells to say, okay, food is in here, we need to secrete more acid. Acid begins to be secreted, the food begins to be acidified. Why is this important? Well, your stomach is acidic, which kills microorganisms. So if you do have a pathogen that you've been exposed to, it has a very difficult time surviving a pH of 1.5. Infants, on the other hand, their stomach only acidifies to a pH of about 3.5. This is why you never, ever, 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 in all your days being alive, give a baby honey. Because that pH is insufficient to kill Clostridium. So if a baby consumes honey, it naturally contains Clostridium botulinum, and that baby will get extremely sick. So you never, ever, ever feed a baby honey, because its stomach isn't acidic enough to kill the natural microflora of some foods. Again, remember when we talk about sterilization, we're not killing all the bacteria, we're only killing the bacteria that's able to grow in that food. So in honey, it doesn't matter if Clostridium botulinum is in there, because it's anaerobic bacteria, which we'll talk about, so it doesn't grow in an aerobic environment. Now, we drink it or we eat that honey, our stomach is a pH of 1.5, it kills the Clostridium botulinum, we're fine. In a baby, that Clostridium, once it reaches the GI, once it reaches the intestines in an anaerobic environment, it begins to sporulate. and it begins to cause a foodborne infection and makes that infant extremely sick. So never, ever, ever give a baby honey because it's not pasteurized. You can't pasteurize honey. So again, that perinatal cells begin to extract acid, and as this is going, we see a mixing. So now what we're doing, we have a camera in the fundus of the stomach looking down into the atrium. So in a second, you're going to see that little black hole. That's the pleura sphincter, so that's the size restriction, about 1 centimeter. And here's an antral contractive wave coming. So this is your stomach churning. So imagine now you have this mixture of food where you've broken it up, the particle size, you've added salivary amylase, so it's being digested, you've got pepsinogen in there, and now you've got this strong contractive wave coming down and mixing and breaking that food up. The other thing it's doing is it's taking it, imagine a bag, and it's squeezing that food against the pleura sphincter. So it's pushing the food out of the stomach into the intestine. So we can interact with that from a food perspective, in a food engineering perspective. We can design foods that are resistant to that mechanical churning. We can add dietary fibers so that force doesn't push the food out of the stomach as quickly. So we can slow things like gastric emptying. Your stomach stays distended for a longer period of time, you're going to feel full for a longer period of time. So when I teach this class, I get 10,000 steps every time, like mid-class, and it distracts me. I hardly ever get 10,000 steps. All right, so again, these antral contractive waves are churning your food. The other thing is when you get hungry, remember we talk about PYY, stimulates gastric motility. Your gastric stomach starts, those antral waves start going. If there's no food in there, you've got this aqueous mixture of secretions, which is low pH, some enzymes, and you've got that antral wave, and now all of a sudden that liquid begins to undergo retropulsion. So imagine you're taking that liquid, you're pushing it through a small orifice, you're going to get a backflow. That's your stomach growling. So you feel that when you get hungry because your hormones are starting to release signals to say, you've got to eat food. Your stomach says, I'm hungry, food's coming, and starts to prepare itself. So it's secreting its enzymes, it's secreting its acid, and it's undergoing these contractions, all to prepare itself for food. Now, in that, again, that stomach, we secrete a lot of mucin. Again, that mucin is to protect that gastric surface from the enzymes that are present. So if pepsinogen is allowed to be active and there's no mucin, it's going to begin to cleave or begin to digest the stomach itself. So we produce a lot of mucin. We produce also intrinsic factors. This isn't that important for the course, but it's important to know. So B12, not bioaccessible. We release an intrinsic factor which binds to B12 and allows it to get absorbed in the distal ileum. So we sometimes release hormones or enzymes that are able to bind with certain nutrients that are able to facilitate absorption. So again, the intrinsic factor is a glycoprotein which interacts very closely with B12, protecting it from the stomach acid, protecting it from digestive enzymes, and it allows it to be absorbed in the terminal or the distal end of the ileum. We also have two other sets of secretions happening, and they're happening in different parts or in different cells. So we have the perinatal cells, which are acidifying the stomach. This is great because it's killing bacteria, but it's also denaturing proteins. Now, why does that matter? Imagine taking a glass of milk and drinking milk. Now, we can think of this either from an adult perspective or, more importantly, from an infant. So we drink that milk, that protein denatures, it unfolds. The hydrodynamic radius of that protein increases. So milk proteins are globular, they're very compact. When they expose to enzymes, those bonds begin to unfold, they begin to become denatured. If they unfold, they begin to become entangled with each other. They increase the viscosity. They slow gastric emptying. They allow more residence time in the stomach. Longer residence time in the stomach means your stomach is distended for a longer period of time. If your stomach is distended for a longer period of time, you're going to have that feeling of satiation for a longer period. Our body is designed to interact with foods. So if you think of an infant and it's consuming breast milk or infant formula, if that stays as a liquid, it's going to very quickly go through that pleura sphincter, right? That pleura sphincter is about 1 centimeter wide. Imagine a bucket with a 1-centimeter hole, fill it up with milk. How quickly is it going to empty? Now that milk goes into the acidified environment, it denatures, it hits its isoelectric point. Its isoelectric point is the point where it carries no net charge. You would have talked about this in first year chemistry, doing your titrations, right? You did your titration, you saw your curve, you saw your drop in pH, and then you'd see your plateau, and you'd look at where the middle of that curve was, and you'd say, that's the pKa of my acid, or, right? You guys remember that? So all molecules have this pKa. Proteins, that isoelectric point means it coagulates. We make tofu based on the isoelectric point. We make cheese based on the isoelectric point of that protein. So when that protein hits the stomach, it denatures. It becomes a solid mass. Milk turns to cheese. I've been watching this clock and I've looked at it about 4 times and I'm like, time is literally standing still. I am so good. And then I realized the clock's not working. All right. So again, we have these 2 separate sets of cells in our stomach. The first set of cells is releasing acid. The second set of cells, the chief cells, are releasing pepsinogen. Pepsinogen is an inactive form of that enzyme, which an inactive form of an enzyme is called a zymogen. That zymogen is produced because if it was active, it would begin to digest itself. It then gets secreted from these gastric pits, moves into the atrium and the fundus of the stomach. The HCl begins to interact with the pepsin, and we go pepsinogen, and we end up with the active site being exposed and allowing that pepsin then to interact with amino acids and begin to cleave amino acids from that peptide backbone. Again, when we talk about the different amino acids, specific proteases will interact with specific amino acids. We'll talk about it in a little bit. But when we talk about the digestion of protein, there's 2 aspects that are really important. The first is that denaturation of that protein. It's unfolding itself. It's becoming a linear form of that globular or that fibril protein. By unfolding it, the enzyme can interact with it and can begin to cleave off the individual amino acids, which will then be available for absorption. Gastric lipase, again, just like the lingual lipase, relies on this histidine, aspartic acid, histidine, serine triad, undergoes the exact same reaction. The amazing thing is, it's a completely different set of amino acids. So the same amino acids, but located completely different spots of that enzyme. So again, those 2 proteins have to fold in totally different ways to get that catalytic triad to form in close proximity, so it can go through those series of deprotonations. It can then attack that carbonyl as a nucleophile. Nucleophiles, you talk in first year chemistry, second year organic chemistry. That nucleophile attacks the carbonyl on that lipid, it cleaves it, the histidine then donates the hydrogen, and again, it cleaves the triglyceride into a 2-monoglyceride and 2 fatty acids. And those can be absorbed. Now, what's interesting here, if you remember, the first slide really was preferential to medium chain triglycerides, the lingual lipase. This one does short and long. Again, when we talk about that catalytic triad being located somewhere on a catalytic site, that molecule has to be able to fit into that catalytic site. And how that molecule fits into the catalytic site is dependent on the length of those fatty acids. So if you have a fish oil with 20 carbons, 25 carbons, or if you have milk fat with something like butyric acid with only 4 carbons, the molecule, the geometry of that molecule is going to fit very differently into that catalytic site. And then that catalytic triad is going to have a different specificity depending on the length of that triglyceride. But when we talk about digestion, it's not just an enzymatic breakdown of food. So what we just consumed, that bread and the cracker, aren't the best examples when we talk about digestion. Think about an apple. when you bite into an apple, when you chew an apple, and when you swallow it, you have very large, dense pieces. Another really great example is an almond. If you eat a whole almond and you chew that almond and you swallow that almond, there's structures in there that are very well preserved. So how do we disintegrate or break up particles of food? So think about when you eat corn, what happens? On the way out, everyone has decorated feces, right? If they've eaten corn. Our body doesn't produce the enzymes to break down that portion of the corn. But think about almonds, think about apples, think about really hard, dense foods that we swallow. How do those begin to be broken down? Typically, it's broken down by the acid. So what happens is the proteins begin to unfold, the structure begins to open up, and water begins to imbibe into that structure. So again, you take an almond, you throw that almond into a beaker of water, come back the next day, that almond's going to still look pretty consistent. Throw that almond into 37-degree water with a little bit of hydrochloric acid, and the structure's slowly going to soften. It's going to begin to be degraded. So this acidic environment is actually changing the macrostructure of the foods, and the macrostructure determines the interface at which those enzymes can interact. So again, if you have a very dense chunk of almond, you're going to have enzymes interacting at the interface, but what about the core? The core of that almond won't be digested. So when you talk about the caloric density of almonds, it's extremely high. You only get about 40% of the calories that's listed on that nutrient label. So there's a group out of UC Davis named Paul Singh that's doing a lot of work with the almond board, trying to rechange how we calculate caloric density. Because in an almond, you don't get all the calories out of it because the structures in which that food is packed, all those molecules designed in those really densely packed nuts aren't able to be degraded as efficiently. So soft foods, things like breads and crackers and soups and gravies, those our body have very easy time interacting with. But when we talk about really hard, dense foods, foods that still have interface, we don't necessarily get all the nutrients out of them. So it depends on how long you chew that food for, how much you masticate it, how you form the bolus, how long it's in the stomach, how quick your stomach empties, and all of that is regulated by the neural impulses of PYY, PP, and PPY. So next day, we're going to pick up here and we're going to talk about a set of hormones that are released from the stomach when it's full. We talk about the hunger hormone ghrelin. So that'll be next day. So I'll see you Thursday.